Thank you very much for the kind invitation. So it's not on the screen. Does it come on the screen? Oh, well, it doesn't matter. I can see it here. Can't see anything on the screen, but it doesn't matter. Well, buenos días. And gracias por la invitación. And eso es más o menos todo lo que puedo decir en español. So today I'd like to talk about two stories that we have in the lab. One is about metabolic intervention in MDS, which is myelodysplastic syndromes, which is published work, and I'm going to tell you what we are doing with that. And another study which is not published yet, and it's about endoplasmic reticulum stress that governs the splicing program in healthy and malignant cells. So I know that we might not need this cartoon here, but I always love to remind people how important are these stem cells, and the fact that we start with only 50 to 200,000 cells at birth, HSC cells, stem cells. And there is someone on my phone. And so those cells are going to produce all the hematologic cells circulating in the blood for your lifelong. And when you think about the fact that trillions of cells are produced every day to renew the cells, and that about 86% of these cells are hematologic cells, far beyond just gut cells or epithelial cells, etc. This is quite impressive. And that requires a very high metabolic and transcriptomic plasticity. Actually, about 65% of the cells which are daily renewed in our bodies are erythroid cells, which highlight, again, this high metabolism. So I decided just to start with a little bit of background. So I've always been working on human hemolytic cells, and more specifically on stem cells. Started by working on the hypoxia-inducible factors, and hypoxia-inducible factors. And what we did was to silence these hypoxia-inducible factors in CD34 from umbilical cord blood, and that triggered. It works. Yeah, thanks. and so when you silence hypoxia-inducible factor, and more specifically the hypoxia-inducible factor 2, which is EPAS1, in CD34 cells from the cord blood, you trigger antioxidant reactions that trigger what we call endoplasmic reticulum stress response, which is lethal for the cells. Then I started working on trying to translate this knowledge of stem cells into leukemia background, and I started studying MDS. So MDS is a myelodysplastic neoplasia, or neoplasm. It's recently changed name from myelodysplastic syndromes to neoplasm. And it's what we call an elderly disease. In 50% of cases, you will find mutations in splicing factors. So splicing factors are these factors that go into edit your pre-mRNA into mature RNA. And about 99% of your RNA are going to be actually edited. So when you have a mutation in one of these splicing factors, it creates catastrophic events which alter most of your transcripts. The question is which one of these transcripts are actually driving the disease. And so we focus on these MDS patients. And for these MDS patients, which are elderly patients, they are usually too frail to receive transplantations. And they are also too frail to receive intensive chemotherapy. And they are left with very few options. So the patient shows with great fatigue, comorbidity factors, and a very strong anemia. So what you can do is to try to rescue anemia with erythropoietin or erythropoietic stimulating agents, which are those growth factors that you can inject. The problem with that is that most of the patients within 18 to 24 months will actually become refractory to the treatment. When they are refractory, the only option left to rescue the levels of hemoglobin, red blood cells, oxygen, are actually transfusion. And the transfusion comes with a very high burden of, actually it's now the greatest risk of death in MDS. The reason is because they get transfusion of 2 units on an average of every 4 weeks. And with that, you are going to build up iron overload and the toxicity which is associated. So when we started working on the stem cells of MDS, there was one question: is actually MDS a stem cell disease? And for that, what we did was to track a mutation in one of the splicing factor, which is SS3B1, and we could track in human cells the origin of the stem cells, and we went as far as the CD49F phenotype, which was developed by the group from John Dick. And we could show that it's emerged and can propagate the disease in the more mature cells, but also in the PDX models. And the nice things with that is that we could follow then the clonal dynamic and show that it mimics quite well what is happening in patients. Now, when I started the lab a few years ago now, I really wanted to understand how these splicing factors are going to drive the disease. They are first hit mutations. They are part of actually SS3B1 is one of these clonal hematopoietic mutations that you can find 20, 40 years before the disease onset. Sorry. And so I explained to you, this is a splicing factor. Splicing factors are here to edit your mRNA into mature RNA. The way they do that is by removing introns and exchanging some of the exons. In the case of SS3B1, one is misspliced, one is mutated. What's happening is neither a loss of function nor a gain of function. It's what we call a neomorphic activity. The reason is because SS3B1 normally recognizes what we call a branch point in the introns that allow the removal of the intron. When it's mutated, it's going to recognize an upstream branch point or cryptic site of splice, which means that when it's going to remove the introns, it's going to leave some nucleotides. And these nucleotides then are now in the frame of your mRNA and are going to create a mess when the mRNA is translated. So patient shows with very strong anemia. So when we did our study, what we wanted to identify are factors that might be linked to actually anemia. And what we found is that COSY, which is a coenzyme A synthase, and PHGDH, which is a key enzyme on the synthesis of the serine pathway and glycine pathways, are actually misspliced. And this missplicing leads to the loss of the expression of the protein. About 50% of the protein is lost for both COSY and PHGDH. Why is it important? It's important because these two enzymes are actually generating the two substrates of the first step of heme synthesis. So succinyl-CoA and glycine are key to generate heme. Without heme, you can't differentiate your erythroid cells into mature erythrocytes. Because with heme, you have the hemoglobin instead. So what we showed with this study is actually that so we show that they are misspliced, that the consequences are decreasing in heme. But what we could show as well is that if you supplement these patient cells with vitamin B5 or succinyl-CoA, which is a byproduct, and vitamin B5, which is a substrate of this enzyme, COSY, you can saturate the system and allow and push the differentiations of primary patient cells in vitro. I'm just going to show one slide on that. So COSY is misspliced. You lose half of the protein about, and then the substrate is vitamin B5 that is converted in CoA. CoA is integrated in TC cycle and also in other mechanisms such as fatty acid, etc. And via the TCA cycle, you get the succinyl-CoA, and succinyl-CoA plus glycine are the substrate for the heme synthesis. So we hypothesized at the time that if you have less COSY, you'll have less CoA, less succinyl-CoA, and less heme and less differentiation. And we could show all of that. Just going to show you a brief summary. We use a cord blood CD34 cells. We transduce them with shRNA against COSY and generating colonies. When you do that, you can see that you have a decrease in total number of colonies, and mainly actually affecting the erythroid compartment. Then you can take the stem cells and do erythroid differentiation in vitro. And when you do that, you can follow the acquisition of CD71, which is a transferrin receptor, when they commit toward erythroid lineage, then 235A, which is a glycophorin A, and eventually it's a kind of a horseshoe. They lose CD71 and keep only the CD235A, glycophorin A staining when they are mature. And so that create different quadrants that you can follow during the erythroid differentiation. And you can see that when the cells are knocked down for COSY, what you observe is an accumulation of CD71, CD235A positive, which are progenitors of erythroid differentiation, and you have less mature cells. And it is very interesting because actually, if you take patient cells, this is exactly the phenotype they have. They are stuck in this CD71, 235A positive phenotype, and they won't fully differentiate. And actually, this leads as well to a decrease in heme, because those are the cells that produce the heme. I mean, you need the heme to have your full differentiation. So very briefly, we use primary patient cells, of course, and what you see here is a protein of expression of COSY. On the left side, you have a healthy bone marrow, so G-mobilized cells. Then you have MDS AML patient samples, which were SS3B1 wild type. And you clearly see that you have different levels of expression of COSY. While if you take patients which have different variants of SS3B1, that are mutated for SS3B1, you clearly see that you lose the protein. And with that in mind, you lose the protein, but you can still see that some of the protein. So we came with the idea that I mentioned earlier, that if you saturate the system with vitamin B5 or succinylCoA, we could potentially rescue some of this erythroid differentiation. And this is actually what we did. We have about 5 patients which are pulled together. And you clearly see that when you add vitamin B5 or succinylCoA to the media, you push the cells to differentiate even further into the erythroid maturation. So the question is, where are we going with that? And so that was a study which was published. Vitamin B5 is a substrate of COSY that generates hemoglobin eventually and red blood cells. In healthy individuals, you have a lot of COSY, you have a lot of vitamin B5, you can generate your red blood cells. This is the end. Thank you very much for that. Am I doing something wrong? So I can continue without the slides. No, there is nothing on the screen. I'm back. In MDS patients, you will have understood you have less COSY, which means that you can process less vitamin B5 and you have less red blood cells produced. And the idea is that potentially if you supplement the diet of patients with vitamin B5, potentially you could enhance the capacity of the cells to produce red blood cells. And why is it interesting? It's very interesting because actually vitamin B5 is a vitamin, of course, has been used in clinical trials, very few, but in some, at very high doses for long time, and it has very minimal side effects. And it shows that you can lower cholesterol, acne, so that's all benefits all in one. And it's important because it's also orally available. So you can just take a pill, maybe in the morning, in the evening, and build up your vitamin B5 and improve your erythroid differentiation. So that's why we are now trying to actually develop a clinical trial that would be a single-arm phase 2 multi-center Bayesian entre-patient dose desescalation to assess the activity of vitamin B5 in patients. So I'm collaborating with Oni Chaudhuri, which is at the bottom left, a clinician in Oxford. Shoshi, that you see on the pictures, a clinical fellow in the lab, working on the project as well. And Rebecca on the right side is the manager of the clinical trial unit that is in charge of all the logistic of patients recruitment and circulation of the samples. So the study design is the following. We will recruit any patients over 18 years old, SS3B1 mutated, because we really want to focus on the mutations before we can extend to potentially other MDS patients that could benefit from vitamin B5. MDS SS3B1 are usually lower risk MDS, and we need a multi-center recruitment, so about 5 sites will be open in UK. if we get it funded. So we'll approach the project with a vitamin B5 dose escalation because we know that high doses are not toxic. So we just want to give one dose very high. If ever there is some side effects which are not anticipated, we'll reduce the dose. And our endpoints are rescuing the hemoglobin levels and hopefully spacing the time where transfusion will be needed for the patients. Then we'll also assess the quality of life of the patients. And the idea is to recruit patients over 2 years, on 5 years or trial. So I'm discussing this one because we are actually applying for funding and we submitted a few applications and hopefully we can have some good news soon. In addition to the clinical trial, the patient samples will be leveraged to generate more data on SS3B1 clones, such as the evolution of the VAF, the variant allele frequency, upon treatment with vitamin B5. Are we changing the fitness of the clone, etc. We also know that vitamin B5 acts on the immune microenvironment and the microenvironment, so we'll be assessing immune profiling, etc. So that's about that on the vitamin B5 story. And I'd now like to switch for the next part of the talk on the endoplasmic reticulum stress that governs a splicing program in healthy and malignant cells. Just to start on what is the ER. So the ER is this organelle in our cells that is involved in the maturations of proteins, but also exposition of MHC, peptides, antigens, and a lot of other mechanisms. And there's really a central sensor in our cells that can signal to 3 different pathways, which are the ITF6, PERK, and IL-1 pathways. The way it works is that you have this chaperone protein called BIP or GRP78 or HSPA5, I don't know, it's so many names, which is actually binding these 3 arms. And by binding these 3 arms, it prevents the activation of these different effectors. What happens is when the ER start to be loaded with unfolded proteins or damaged proteins, BIP is going to dislodge from these factors and bind, because it's a chaperone protein, it's going to bind to these unfolded proteins, and that allows those 3 arms to be activated. So the ITF6 is actually going to translocate to the Golgi apparatus, where it will be cleaved, then it will be cleaved in the cytoplasm to generate actually a transcription factor. PERK is a very interesting one. It's a kinase, and they are going to dimerize to autophosphorylate themselves and phosphorylate its main substrate, which is eIF2 alpha, which is a main, which is actually the initiation factor of translation. When eIF2 alpha is phosphorylated, it blocks the translation. So it's all in the logic of trying to rescue the stress that you have in your cells. You have unfolded proteins, which are loaded in your ER. You don't want to load more, so you prevent mRNA from being translated. And for that, you phosphorylate eIF2 alpha. What is funny as well and very interesting is that some proteins are still translated. To be translated, they need specific sequence that are either upstream or IRS or the 3' media that allow non-conventional translation, such as ITF. And then we have IRI1. IRI1 is a dual kinase and ribonuclease. And it can do this unconventional splicing. So splicing, we understand, is a pre-mRNA matured in mature RNA. And normally it happens in the nucleus. But in this specific case, we know that XBP1, which is key for many diseases, is actually spliced by the ribonuclease activity of IRI1 to generate a mature RNA, which is quite stable and leads to the translation of a stable protein that is also a transcription factor. And there are many stresses that can activate these pathways, including starvations, hypoxia, inflammations, infections. And that signals through the ER stress, which is called also UPR for unfolded protein response. Or actually, there is another very similar mechanism called ISR for integrated stress response. And all of that is with the idea to allow the cells to recover by generating more folding capacity, antioxidant capacities, mRNA degradations, protein degradation to avoid to saturate your system. If the cells are not capable of doing that, what they will do is to trigger apoptosis, this kind of on-off system. If you can't fix it, degrade it. So, that was for the ER introduction. When we started the lab, some time ago now, we had these questions about proteins. So in the lab, we do a lot of splicing and a lot of proteostasis in hematopoietic cells and leukemic cells. And we had this question about, can we capture proteins which are specifically translated upon stress? So what we call de novo synthesized proteins. We benchmark a little bit what was done around. You have SciLac approach where you use heavy chain, light chain, etc. It requires specific media, which is a bit heavy to do. Then you have indirect approach, which will be ribosic, so ribosome sequencing, where you sequence whatever is stuck in your ribosome, which should be a picture of what was translated at T time. And there was this technique, which was started to be published at the time, where you can use O-propagate puromycin, which is an analog of puromycin. It's going to be integrated into the nascent chain of peptides. And when it's integrated, it stops the elongation. And this is very interesting because this OPP can just be added to any media. You don't have to have a specific media to use or medium. You can even use it in vivo. The only thing to keep in mind is that you stop the elongation. So it's puromycin. So if you don't want to induce a stress, you need to make sure that the time you are using it is not inducing any proteostress in your cells, which is not the case at the concentration and the time we use it here. And so once your peptides are labeled, you can pull them down and do some mass spec, which allow you then to discover which are the proteins. What is the advantage of this technique? The advantage of this technique is that when you do mass spec, usually you get the most abundant proteins captured, but you can miss the one which are actually low abundant. With this one, we are going to capture proteins which are induced upon the stress that you will have chosen and that have a high fold change. It doesn't matter if they are highly expressed or not. They will have a high fold change between the time of treatments and no treatments, and even if they are low abundant, and you can capture them. So, mentioned earlier, we're interested by proteostress. And so we induce ER stress using tunicamycin, which is an N-glycosylation inhibitor, but which is a very well established inducer of ER stress. And so what you do, you treat your cells. You can do that for 4 hours. After 3 hours and a half, you just add your PP to your media. Half an hour later, you stop everything and collect your samples. We did that. And what we capture is about 350 proteins, which were synthesized upon de novo translation, upon stress with ER stress. So what you see, each column is actually 1 dose of OPP because we wanted to show that we capture the peptide and we can saturate the system. So we have doses of 0, 0.5, 1, 2, 1, 5 macromolar. And like this, you capture proteins. Then with proteins, it's like gene, and you can do gene ontology, or protein ontology. And you, of course, have pathways involved in protein catabolism and ER stress, IRA1, etc. But what we observe is that we actually have a lot of pathways which seems to be involved in mRNA metabolism, such as splicing. So you will find a lot of helicases, RNA binding proteins, splicing factors, which are captured by this approach. And this is very interesting because at the time when we were starting doing these experiments, we were working on the SS31 paper, so which is a lot of splicing. And also there is nothing known about ER playing a role in splicing and metabolism. So for example, if you look at SRSF1, that is on the table, this is the mass spec data. So you can see that with different concentration of OPP, you capture them, and so you have 0.5, 1, 2 micromolar. From 2 micromolar, you reach a plateau. So you have captured all the SRSF1, which was translated between these 30 minutes. Then you can do Western blot, and you can show that it's not just true on the mass spec. You also see it when you induce ER stress. Actually, you see the hyperphosphorylated band of SRSF1, which is a sign of activation. And the question is that, is it actually a transcriptional regulation, although it's very short term because it's only 4 hours, or is it more translational stabilization reaction? And so with some qPCRs and when you look at the expression of SRSF1, at least at 4 hours, we don't have any increase. If anything, it's more decrease. So it's likely that this activation is really translational dependent more than transcriptional dependent. But when you talk about splicing, what you need is actually to look at the RNA. And what we did is to use OCIML3, the leukemic cell line, also HEC290T cells, because at the time we were already thinking that maybe it's not just a hematopoietic system, but more something that will have a physiological role. So here is gene expression with a heat map. So you have the HEC290 cells and the OCIML3 induced with tunicamycin, the ER stress. And you clearly see the change in gene expression with all the classical ER stress activated. What you see on the graph is what we did is we plotted the proteins which were captured, or the gene expression here, on the RNA-seq, just to show that actually, apart from CHAC1, which is a top left in green here, that is a redox protein, nothing to do with splicing. It's regulated at the transcriptional level and is captured by the proteomic and is a non-target of ER stress. The other one seems to be on the flat curve, so it doesn't seem to be regulated at the transcriptional level. So when you have RNA-seq data, what you can do, if you have enough depth of sequencing, is to analyze the splicing. And the splicing, just in brief, it's categorized according to the type of splicing. You have skipping exons. So skipping exons can be misleading, but skipping exons doesn't mean necessarily that you just skip things. It means inclusion or exclusion. So either you keep or you remove the exons. Then you can have this 3' splice site where you leave a bit of nucleotides at the end of your introns, 5' at the beginning, mix exon exclusive, and intron retention. And so on the RNA-seq data, we analyze the isoform, and clearly you see that upon ER stress activation, you have a switch in isoforms. And this isoform were mainly skipping exons. And that's true for the OCIML3, it's true for the HEC290 cells, and we use 2 time points of OCIML3, we use 4 hours and 24 hours. So now we have this RNA-seq data, and we want to understand whether there are any common transcripts which are spliced at the different time points and in the different models. So you look at the genes which are have the skipping exons that are captured in skipping exons, and we observe 102 genes which were spliced. So when you talk about splicing, your gene or transcripts can be spliced at different sites. So out of these 102 transcripts which were spliced and common to the HEC290 cells and the OCI at 4 and 24 hours, actually you have 33 which have exactly the same splicing happening in HEC or in OCI. So for example, PRR3, that you see here, is one of these transcripts where the exons is exactly the same splice in OCI or HEC290. So to avoid to have to do RNA-seq each time we wanted to see if we had activation of this mechanism, what we did is to look into the 33 events and check for transcript where we could design primers and capture these events. And that's why you see PRR3, PS3, REH2, and RBM39. Sorry. And so what you can see now, because now we can design primers, and you clearly see that upon tunicamycin, you have this isoform that appears. Now that we know that, what we can do is also design primers that are not just in exon 2 or 3, but are overlapping between 2B and 3. which means that by qPCR, you can then assess if you see the splicing out. So when we activate ER stress now, we clearly see a switch in splicing, which again is not known. There's nothing about that. So what is the impact of splicing? The impact of splicing is actually very difficult to determine, at least in silico, and you need really to have approaches which are looking at the effect on the proteins. And so, for example, if you look at RBM39, we see a splicing as soon as 4 hours, but you can clearly see that at 4 hours, you don't really see any effect on the protein. You see it later. For AP3S2, we have a very nice splicing, but it doesn't seem to affect really the protein. It could affect the localization of the proteins or other functions, but here is not clear. So what I want you to keep in mind is that we are only using this one as markers or biomarkers of ER stress activating splicing before investigating the function value of these targets. Again, just to show you, it's not really regulated at the transcription level, not at 4 hours, maybe at 24 hours for AP3S2 and RBM39. That might explain what we see here on the protein level. But at the early time point, it's really not a transcription impact. So then we come back on the ER stress, because now we show that ER stress seems to activate some splicing events. I'm sorry, just before. Now that we have the markers, we can really investigate that in many cell types. And we use hepatic cells, PC3, MCF7, breast, HeLa, and even primary MSC cells, which are mesenchymal stem and progenitor cells. And 1, 2, 3 are 3 different patients, donors. And you clearly see that each time you activate ER stress with tunicamycin or tapsigargine, which is another inducer of ER stress, you clearly have this phenotype. And so the question is actually which arm of the ER is driving this phenotype. And I mentioned that there are 3 arms, and the nice thing is that there are inhibitors for these 3 arms. And what I'm going to show you is color-coded now. So in black is DMSO, in red is tunicamycin, so we use the splicing with the qPCR on the target. And then in green, inhibitor of PERK, in orange, inhibitor of IRE1. And what you see is actually that it doesn't seem that the ITF6 pathway is driving the phenotype, because when you inhibit it, you don't rescue the splicing, but the SHOP and the PERK and IRE1 pathway seems to be involved. The problem with this inhibitor is actually that they have some redundant activities. For example, its PP1 is a target of IRE1, but if you use a PERK inhibitor in green, you inhibit it as well. And SHOP is a target of PERK, but if you use IRE1 inhibitor, you inhibit it as well. So now that we know it's either PERK or IRE1, what we did is to generate a genetic approach where we have vectors which are DOX inducible to activate either the PERK arm or the IRE1 arm. So it's just a validation that you see here. But now we can do again, treat our cells with DOX, which express the PERK, or DOX, which express the IRE1. And you clearly see that when you activate PERK, you see the splicing occurring, while if you activate IRE1, you don't see much happening. So now we have another data set of splicing that we can combine with the first one. Again, what we are trying to do here is, because it's a new, potentially new mechanism, we're trying to see if it's true, and if it's true across tissues. Then if it is true, the impact that ER stress will have on splicing might depend on your mutational background, but you will still have a few core genes which are always spliced, kind of a signature. So here, the nice thing is that we have a pharmacological approach with tunicamycin. We have different tissues, different time points, and then we have the MSCs, the primary human cells, and it's a genetic approach. And we exclude the IRE1 to avoid redundancy that can be induced by pharmacological inhibitors. And now, out of the 102 transcripts which were earlier spliced, you have 38. Out of the 38, you still have 8 which have exactly the same coordinate, which means that PR3, PS3S2, REH2, RBM39, and NUP, and EIF will have always the same exons, because here we're looking at exons, splice. They can be either excluded or included. For example, the I4, I2, it's an exclusion. So we know that PERK is a kinase. So to try to go upstream or downstream in the pathway, we generated some phosphoproteomic analysis. And we did that as early as 4 hours, again, 24 hours. And what we observed is actually that you have a lot of phosphocytes which are involved in protein folding. It makes a lot of sense. It's ER induced. You also, again, have a lot of RNA binding proteins that seems to have phosphocyte activated. And if you have splicing factors, sorry, if you have splicing factors have specific domains which are usually rich in serine arginine, so SR proteins, and you can run phospho-SR Western blot to see if you can validate the phosphoproteomic data and see if you can see these phospho sites activated. And this is exactly what you are looking at here. We did that in OCI, MS3, and in MSCs, again, primary cells versus leukemic cell lines. And clearly, you can see that when you activate ER stress, as soon as 2 hours and 4 hours, you have this phosphorylation of splicing factors, SF4, 5, 6, and 1. And so when you activate ER stress, you have activation of different splicing, and it seems to potentially be driven by phosphorylation of splicing factors. So just to make sure that it was PERK driving that, and you clearly see here that you have 2 hours already some signal. So what we did is to redo the same experiment, but with this time with at 2 hours with tunicamycin or with a PERK inhibitor. And so with tunicamycin, you see the phosphocyte increasing, and with the PERK inhibitor, you rescue this phenotype. So it seems that PERK is inducing phosphorylation of splicing factors. We know that it's unlikely that PERK directly phosphorylates splicing factors. The reason is because we know actually that there are families of kinases which are especially designed to phosphorylate splicing factors, like the SRPK families and CLK families. And so the SRPK are usually involved in the shuttling of splicing factors from the cytoplasm to the nucleus, and the CLK in the activation of splicing factor in the nucleus. And they are nice inhibitors for all of them. So we redid the same experiment. This time we activate ER stress, you see the splicing. You use a CLK inhibitor as SRPK alone, there's nothing much. Now you can use tunicamycin plus CLK inhibitor. You clearly see that you prevent the splicing. And you can use SRPK inhibitor and you nothing happen. So it seems that CLK here is driving the phenotype. How is CLK driving the phenotype? That was so we went back on our phospho-SR proteins, and then we actually generated a Western blot on CLK. And what is happening is that as ER stress is activated, CLK is translated or stabilized. And as soon as 2 hours, and that's why you see this phenotype as well. So this is nice, but so far what we have been using are pharmacological inductions or genetic inductions. We were wondering if it actually was true in real data. So we generated an ER score, which is just based on the expression of genes which are very well established as ER targets. And we run it through the TCGD data, and you can see each dot is a patient. And so which means that you have different levels of ER across the different groups of patients. And then what we did is that with the Broad Institute, actually, you can access the exon count. And just to explain, so we have the ER score, which is a gene expression. And then we have generated an ER splice signature where we look at the exons. Is the exon present or not? And the idea is that if ER stress is driving this ER splice signature, we should see a correlation between ER score and ER splice. And to cut a long story short, we actually can see in breast cancer, pancreatic, prostate cancer, this one where with a high number of patients, You can see a slope. It's slow, but it clearly is significant and significant in the different cancers. And if you take the different quartiles, the bottom one and top one, you can actually see that it's even stronger. So what we show here is that when you activate ER stress, you actually translate some splicing factors and you activate CLK, that phospholipid splicing factors, and it's going to drive a whole new phenotype of splicing in your cells. And the question now is, first, what is it used for? I can tell you that if you inhibit CLK and activate ER stress, you have a synergistic effect in killing the cells. And it's also true for RBM39. Then it will depend on what gene expression you have in your cells, because you can command the splicing, but you don't command the gene expression. So gene expression will be driven by your different oncogenes. So this signature might be different in different cell type and the impact on the cells as well. And on that, I would like to finish and to thank the people who did the work. All listed here, Céline Phillips, that actually led both stories, the vitamin B5 and the splicing, and just got a position of independent researcher in France. The patients with Carreras samples and the different sponsors and their charities. And you, of course, for your attention. Thank you.
Thank you for this amazing talk. I would like to open the room for discussion. Any question?
Hi, Kevin. So thanks a lot for that. I very much enjoyed both parts, but I would like to ask about the first one.
Yeah.
So is there more metabolic enzymes dysregulated in SF3B1? And can we explore that even more by maybe proposing tailored diets?
Yeah, and they are actually. PHGDH, which is a serine metabolism, and it's very interesting as well. The difference, the reason we focused on COSY, so on CoA metabolism rather than glycine metabolism or serine, is because the serine and glycine can be come from the microenvironment themselves, while CoA are not permeable and has to be produced within the cells. But definitely there is things to do. When we think about the glycine, for example, it's key in the one carbon metabolism, which means that is actually potentially key as well in SAM. So the methionine, which is a donor of methane for methylation. And you can potentially think that you will have a remodeling of chromatin through different methylation, and also the same thing with CoA. CoA with acyl-CoA is a donor of acyl for acetylation. And so, yes, there's a whole field to explore, I think, and with a metabolomic intervention. Just the only thing, the reason we can't exactly do a direct mouse model SF3B1 and try these different diets, that splicing is highly dependent on the ribonucleotide sequence. And there is quite a few discrepancies in mouse and human in the RNA sequence, and which often shows different splicing. So you won't necessarily see COSY, for example. So most of the main human splicing that we believe drives the disease are not seen in mouse models. So you need to target each single one. But yes, metabolic intervention will be, I think, the path to follow.
If I can continue these questions. I was like wondering if since metabolism is really dynamic, if it is if the cell, well, the cells have reprogrammed to find these metabolites from other pathways, if there is known or if you know.
So the way we did the experiment is that there are a lot of people working on SF3B1 and have produced amazing work. And we wanted to discriminate ourselves a little bit from them. So what we did is to generate colonies, so basically to try to assess functional cells. So we took patient CD34 cells, generated colonies, and we did the splicing analysis on the colonies with the idea that we will identify things which are necessary for the process. And what you see actually when you do a GSEA analysis, or gene set enrichment analysis, is that you have pathways such as Heme, which are highly affected. So you clearly see that the cells are trying to compensate the mechanism. So yes, do they manage? It's another question. We'll try to for if we go further in the reflection here. So SS3B1 are usually low risk MDS, but they can transform. When they transform with very specific oncogenes. And the question then is, what are these oncogenes bringing that is likely to be metabolomic changes that actually allow the fitness of the clone to increase to become transformed into it. And although they are low risk MDS, when they are transformed SS3B1 AML, they are very, very bad prognosis.
Thank you. Any other question?
Hi, hello. Really nice talk. I love it. The last part, I think that I lost some of the results, but I have a question regarding that. Did you find any difference among the different ear stress and the kind of splicing? Like the question will be, there is any difference among of the stress that you generate and the splice that you observe?
So, ER stress, I'm not sure I get the question, but as long as ER stress is activated, so the response, which is the UPR, for the protein response, you have the same splicing occurring. So it's really the same splicing factors which are translated and the same events which occur, which if you, for example, activate ER stress, not ER stress, but via the ISR, which is the integrated stress response that signals through different kinases, but still through the phospho-EF2 alpha, you don't exactly have the same response. And we believe that is because not the same splicing factors are translated. So depending on the stress, if it's ER stress, it's ER stress. So the pathway signal. If it's ISR, we don't get exactly the same results. We get some signature, but not exactly the same. So we believe, yes, indeed, depending on the stress, we might have different response. Then you can think that, again, yeah, what we are trying to do is to demonstrate that it's a true mechanism that is physiologically relevant. But the one we extracted are very specific and they're common to everyone. What you can think is that depending on the tissue you will have specific splicing that are important for this tissue. Depending on the oncogene, you will have specific gene reprogramming and splicing occurring that will be relevant to the disease, more than just the targets that we are showing you.
Thank you.
Any other questions? No? Hello, thank you for your talk. I also have a question for the first part, the MDS part. So if I understood correctly, you said that SF3B1 is one of the early events and it happens 20 to 30 years before the disease.
Yeah, it's one of the clonal hematopoietic mutations like DNMT3A, ISXL1, and TET2. So it's the first one actually coming up.
So I was wondering, and then if I understood correctly, the hypothesis that the phenotype, well, the disease is caused by misplicing of, for example, these two, COSI and the other one. So I was just wondering, why does it take so long? Why does it take so long for the phenotype to be observed?
That's a big question in the chip field. So clonal hematopoiesis with undeterminate potential. Why do you have these myeloid mutations and why are you not transforming? I can give you my own interpretation, but that's my own. And I will highlight it just with one example. So like for example, if you take TET2 clones, so TET2 clones are kind of protected from inflammation, so they don't respond anymore to inflammation, at least partially, which means that if you keep them over time, the clone which is TET2, while the healthy hematopoietic stem cells are mobilized upon inflammation, upon infections, your TET2 clone is just going to keep growing and not exhaust. And you come to a point where mutations are stochastic, but they will happen more frequently in the largest clone. And eventually, with the addition of second hit mutations in TET2, DNMT3A, SS3B1 clones, you will transform your cells, potentially one explanation.
Some other question?
Hi, Kevin. I really enjoy the talk. I just wonder regarding the clinical trial, will you use as an inclusion criteria the allele expression of the wild type form of the SF3B1, meaning that maybe there is some patient in which they have a lower expression of the wild type, but could be a confounding variable?
Yeah, I mean, it's commonly done to track the VAF of, and you know that most of the mutation are heterozygous. So yes, you have the wild type. I mean, we won't use the wild type allele of the mutant clones. Huh? We'll see if we can, if healthy cells, which are still some of them at least present, can outcompete the clone, even if we see a change in the balance of the clone. So if you have 40%, so 80% of your bone marrow, which is malignant, can you see a decrease in these malignant clones and the healthy compartment to come back? So that will be the reduction of the VAF that will indicate.
Hello, it was a nice talk, sir.
Thank you.
I always think of one thing that in terms of immune cells, suppose T cell, when they get activated, they express a lot of proteins, and that might induce the stress in the T cells. So do you think the immune cells also take up the similar kind of machinery like the cancer cells to deal with the stress, because most of the time they don't die and they're always fighting the infection inside our body, and how do they deal with it?
No, that's a very good question. I think we don't know. We don't exactly know how they can have a threshold of stress. So, for example, if you take B cells that produce antibodies, they have a very high ER stress, and so actually any secretory cells will have a very high ER stress, but also a very high tolerance to ER stress. And funny enough, actually, hematopoietic cells have a higher tolerance usually than adherent cells to ER stress. So you can use doses which are higher to activate ER stress that will potentially kill other cells. So we don't know. We don't know how it's the signaling is dampened. I saw this paper recently from here about CBP alpha and the different isoforms that limit the ITF4 program. That could be one of the explanations. It's a nice work, by the way. That would be one explanation, but we don't really know. So that's one question. And I like the idea that it comes from another isoform. Let's come back to the splicing side. We don't know, but I think once we know, I will change our view on a lot of things. And for example, multiple myeloma, where we know that CXBP1 can be mutated, that can be a new angle to push even more strong proteostress.
Thank you.
Here we go.